So it is time to start.
Okay, sir.
Hmm.
So, my voice is audible for everyone?
Yes,
Okay.
you're audible.
Uh, Good morning, all of you. I am Manjusha Pal, and today my presentation topic is non-antibody based proteomic techniques. So, before start my today presentations topic, I am going to introduce about in very brief manner that what is proteomics and which techniques are used in the proteomics. So, moving to the next slide. This is the this is an overview of proteomic techniques. So, in the study of the proteomics, in the techniques in the proteomics, it is a study of the protein in the large scale study, or we can say that in another manner, like it involves the applications of techniques for the identifications and quantifications of overall protein in a cell or a tissues or a or an organisms. When we talk about the techniques which is used in the proteomics, it can be classified in various manners, such as conventional techniques, advanced techniques, gel-based approaches, quantitatives, and high throughput techniques. In conventional techniques, it include the chromatography based techniques like paper chromatography, thin layer chromatography, and HPLC, and ELISA and Western blot. And in advanced techniques, it include the protein micro array, and the gel based approaches is like the electrophoresis. In the electrophoresis, it consists the SDS, agarose gel electrophoresis, and 2D gel electrophoresis, and another one is the mass spectrometry and advanced sequencing. And in the quantitative techniques, it include the ICAT, the SILEX, and ITRAX. And in high throughput techniques, it include X-ray crystallography and NMR spectroscopy and so on techniques. Uh, so see, in particular in one session, it's quite difficult to elaborate all the techniques. So according to my today's presentation topics, I am going to explain the gel-based approaches and mainly focus on 2D electrophoresis and some quantitative techniques like ICAT, SILEC, and ITRAC. This is the next slide. It is a gel-based proteomics. In gel-based proteomics, we use the electrophoresis concept to offer information on protein properties such as the molecular weight and isoelectric point. Actually, it is the most popular and the cheaper approach to screen the protein expression at large scale study. In these techniques, the two-dimensional gel is widely used methods which provide the high resolutions proteome separations. So before explaining the two-dimensional gel electrophoresis, I am going to explain about brief about the electrophoresis. So moving to the next upcoming slide, this is the electrophoresis. So in 1937, the Annie Tislius was first introduced the term electrophoresis and was regarded as the father of electrophoresis. In electrophoresis techniques, the two term comes the electro, which means the energy of electricity, and phoresis, which means the separations. So it is a techniques which used to separate the biomolecules like DNA, RNA, or protein, and according to their size and the charge. Basically, in electrophoresis, it involves the migration of the charged particles or the solutes of any size in a liquid medium under the influence of an electric field. Based on their size and charge, the molecule will travel through the gel in a different direction at a different speed. Like the positively charged particles, which moves to the cathode, and the negatively charged particles, uh, it moves to the anode. So moving to the next slide, this is the factor affecting electrophoresis. The rate of migrations of a solute in an electric field depend upon the following factors, like net charge, size, shape of the particles, the pH of the medium, the strength of the electric field, and the properties of the supporting mediums and the temperature. So these are the certain factors which affect the rate of migrations in the moving electrophoresis. Now I'm moving to the next slide. See, here we can see the components of electrophoresis. Basically, it consists of the 5 components like electrical power, support medium, buffer, and detecting system. As we can see in the figure, it's depicted the electrophoresis apparatus. This is the electrophoresis apparatus, and this is the power supply. So electrophoresis apparatus with the electrical supplying system, and this is the electrophoresis, sorry, this is the electrophoresis tank in which it contains the buffer solution. See, the buffer is a very important because buffer consists electrolytes, and it is a electrolyte solution which carry the current and it maintain the solution of the pH. After applying the electrical supply, it is provided by the electrode that may be the anode and cathode, and sample this is the sample wheel over which the sample is applied, and this is the directions of movement. This is the directions of movement of sample. And so if the samples having the positively charged particles, it means the cations, it moves to the cathode, and the samples which carry the negatively charged particles, it moves to the anode. So moving to the next slide. Depending upon the nature of the support medium and the mode of the techniques, it classifies in a various types of electrophoresis like agarose gel electrophoresis, polyacrylamide gel electrophoresis, cellulose acetate electrophoresis, capillary electrophoresis, and high electrophoresis, and pulse field electrophoresis, and immunoelectrophoresis, and two-dimensional polyacrylamide gel electrophoresis respectively. Now moving to the next slide. Now I'm explaining the two-dimensional gel electrophoresis. In 1975, PHO Farwell was the first to report the 2D gel electrophoresis. In this techniques, the separations of protein is mainly based on the two factors. The first one is the isoelectric focusings, and the second one is the molecular mass. In isoelectric focusings, which separates the protein according to their isoelectric points. And uh, what do you mean by the isoelectric points? So basically, isoelectric points means the point of the pH at which the protein carries no net charge, means the protein becomes zero. And the second factor is the molecular mass, which separates proteins according to their molecular weight. Now moving to the upcoming slide, this is the principle behind the isoelectric focusing. In isoelectric focusings, we use isoelectrical focusing strips of a different pH. And mainly isoelectric focusing is done into a flattened gels. So from the figure 2, we can see that in isoelectric focusings, gel electrophoresis, there is the use of ampholytes, and the ampholytes are used to create the pH gradient. So in the figure 2, this is the pH gradient depicted 3, 4, 5, 6, 7, 8, 9, 10, and so on. So from the pH 0 to 40, the gradient is going to be set up. And after that, we load the sample and electric current is applied and allow the sample to run. Uh, Now in isoelectric focusing, what's going to be happen after the load the sample? as we know that the proteins are made up of the amino acids, and amino acids are zwitterions, which means it carries both charges, negative as well as positive charges. So basically, the protein that are made up of an amino acids is also having the isoelectric point. So when a protein reach in the zone of the pi, at that time, that protein they are going to retard at zero, and they are not going moving further. So here from the figure uh, 3, one concept we should keep in the mind, that when the the proteins of the pi value is high and pH is low, then protein carry positive charges, and when the protein pi value is low and pH is high, then protein carry the negative charges. After the completions of isoelectric after the completion of the first dimension, the isoelectric focusing strips is run perpendicular to the first run along the SDS page gel. After that, in the second dimension, wherein the proteins with similar pi are further separated on the basis on their molecular weight on the SDS page gel. Moving to the yeah, and this is the steps. The first step is the sample preparation. The next one is the separations. The first separations, which is done by isoelectric focusings, and the second dimension separations by the SDS space. And the next steps is the visualizations of the results, and the next one is the further analysis. So the first steps is the sample preparation. So in the sample preparation, the sample needs to be converted to a physiochemical state, and the procedures of the sample preparations are dependent on the sample type. And the next step is the separations by isoelectric focusing. So I already explained how the protein mixtures is loaded in pH gradient and how it migrates. And the second is the SDS space, and in this type, and in this the protein gets separate according to their mass. The next step is the visualizations of result. uh, so in these steps we use multiple methods such as chromatin blue, silver staining for the visualizations of proteins, and after that protein can be separated and informed such as the molecular weight, its pi, and the amount, which can be determined by further analysis. So this is the applications of two-dimensional gel electrophoresis. So basically, as a separations, detectations, and quantification technology, two-dimensional gel electrophoresis is an important technique, especially in especially for the clinical lab. It is involved in the determinations of the protein expressions labels and biomarker discovery. It also provides the direct visual it also provides the direct visual confirmation of charges in protein medications, and it also used for protein fingerprinting of Indian black gram varieties. Now moving to the next slide, the next techniques is the MALDI-TOF MS. MALDI-TOF MS, it is a mass spectrometry method. So I'm going to explain in very brief about mass spectrometry, because we already discussed in our previous sessions by our colleagues, Ms. Totrina Das. So basically, mass spectrometry is an analytical technique that uses the mass to charge ratio of the analyzed sample to determine the amount and the type of chemicals compound which exist in the molecules. So the mass by charge ratio measurements are taken on a molecule in the gas phase, and heating or by the other treatments it required to transfer the macromolecule to the gas phase, which usually results in a very rapid decomposition. Although mass spectrometry has been in use for many years, but it couldn't be applied to the macromolecules such as the proteins and nucleic acids. So that the Kals and the Hillenkamp improved the technique based on the concept of the soft ionizations. As a result, the first matrix assisted laser desorption ionization was developed. Moving to the next slide, this is the MALDI. Actually, MALDI and TOF, these are the two different terms. The first is the MALDI, which considered the ion source, and its abbreviations is a matrix assisted laser desorptions and ionizations. And the second is the TOF, this is the time of flight, which considered the mass analyzer. So basically, it is a type of soft ionizations in which a laser is used to strike a matrix of the tiny molecules and causing the analyte molecules to enter the gas phase without being fragmented or decomposed. MALDI can be used to examine the biomolecules such as the peptides, lipids, sugar, and other organic macromolecules. So moving to the next slide, this is the mechanism behind the MALDI. The mechanism of MALDI is based upon the three basic steps. The first is the formation of solid solution. Next is the matrix excitations, and the third is the analyte ionizations. And this is the principle behind the MALDI. So as a organic substance which containing an absorbent solution which is termed as the matrix, and a matrix is mixed or coated with the material which is going to be examined, and when exposed to the laser light, the multi matrix act as an energy absorbers. See here, the commonly used matrices are the cyano 4 hydroxycinnamic acids, 2,5-dihydroxybenzoic acids, and 3,5-dimethoxy 4 hydroxycinnamic acids, and 2,6-dihydroxyacetone acetophenone are some of the more commonly used matrices. On the target plate, it is composed of conducting metals, and the matrix and analytes are the deposited, and then the sample is then ionized on the spot where it gets excited, and by using the laser beam such as the infrared or UV. So from the figure 4 we can see this is the target, sorry, yeah, this is the target plate, and the here the spot green color shows the matrix spots, and the orange color shows the analyte spot. So basically, what is happening? So on the target plate, it is composed of the conducting metals. The matrix and analyte are deposited, and the sample is then ionized on the spot where it gets excited. This is the laser beam, like UV or infrared. So using a laser beam, and in the presence of the laser beam, the matrix molecules energetically dissolves and ionize, and releasing the analyte molecules into the gas phase. So on the basis of their mass and the charge ratio, the protonated ion accelerates and separates and becomes separates. So basically, a smaller ion molecule accelerates more quickly than the larger ones. And after that, sorry, sorry, yeah. After that, then using a time of flight, which is the analyzer, and these protonated ions are identified and measured. So this is all the principles of MALDI. Now, moving to the next slide, this is the principle of TOF. The basic principle of the TOF analyzer is that it determines the time which is required by the ions of different size and charge ratio, which takes to travel the length of the flight tubes. The ion starts traveling at the same time, where the small and the a lighter sized ion travel fastest to the detector than the larger and the heavier one. So with the use of uh, with the use of only use of the linear TOF analyzers, all ion do not feel the pulse with equal intensity. So therefore, the reflectors is applied. So basically, two types of reflectors. First one is the linear of analyzer, and the second one is the reflectors of analyzers are used. And these two analyzer will shown in next slide. The reflectors contains a high voltage series of a ring electrodes, and it consists a slightly displaced angle that helps the ion to pulse back in the flight tube. So TOF is like it's like a tube like structures, right? Then the ion that start the traveling at the same time, with the small and the lighter sized ion travel faster to the detector than the heavier one. So with the use of so the ions that travel faster will spend more time within the reflectors, where the detector receives similar mass ions at the same time. So basically the TOF analyzer is high in resolution than the other analyzers, and it's applicable only for the microbiological purpose. So this is the two types of reflectors. The first one is the linear TOF analyzers, and the second one is the reflector TOF analyzer. Reflector TOF analyzer, which is basically this is not suitable for the analytes that are not stable enough to survive the electric fields. Now moving to the next slide. This is the diagram of the process of MALDI-TOF mass spectrometry. So basically, these are the these are the samples, and these are the samples, and these are the matrix. So the analyte solutions and the matrix solutions is combined together, and this is the MALDI-TOF MS sample plate. So the mm, molar matrix to the analyte ratio is generally adjusted between the 1000 is to 1 ratio, which is good for optimum MALDI spectra. After that, the mixture is coated onto the metal target plate here for testing. The sample and the matrix mixtures gets co-crystallized after the drying, and it forms a solid deposit. And after that, this sample plate is placed into the MALDI-TOF equipment and evaluated using the software which is specific for the system. And both the sample and the matrix are sublimated and ionized by the MALDI. And after that, these produced ion are separated by the M by Z ratio, M by Z, using a TOF analyzer, and the mass spectrometry software generates and analyzes the spectrum which represent of these ions. Here, this one, resulting in the MS profile. This is the pic of the ions. So moving to the next slide. This is the advantages and the limitations of the MALDI-TOF mass spectrometry. The advantages of the MALDI-TOF mass spectrometry, it is a very highly sensitive method, and it gives a more rapid and accuracy result, and it's a cost saving, and it's a very high throughput and the efficient way in the identifications techniques for both the bacterials and the fungal strains. But the limitations of this technique is that it's a very low analytical sensitivity. It means it not going to distinguish between the two specific bacteria, suppose E. coli and some related families. Now, moving to the next slide, this is the applications of the MALDI-TOF MS. MALDI-TOF MS is has a simple operationals and a good mass accuracy, as well as high resolutions and sensitivity. Therefore, it has widespread uses in proteomics in the field of detections and identifications of the clinical samples, in peptide mass fingerprinting, in MALDI imaging, and oligonucleotide analysis, and in the field of microbiology. So this is all about the MALDI-TOF MS. Now moving to the next slide, the another technique is the ICAD. It means isotope coded affinity tags. It basically label the amino acid based labelings. So ICAT expanded the proteins range to be analyzed and allow the accurate quantifications and the concurrent sequences and identifications of individual proteins in a complex mixture. It is a gel-free methods. Coming to the next slide, this is the components and this is the reagent. I This is the components and this is the reagent. Basically in the ICAD, the three elements or the three types of reagents are used. The first one is the reactive group, the second is the isotypically coded linker, and the third one is the tag. The first is the reactive group. In this type of a group, a specific chemical reactivity group for labeling an amino acid side chains towards sulfhydryl groups. Examples: iodocetate amide to modify the cysteine residues, and the second is the isotypically coded linkers. Examples: deuterium, and the third is a tag like biotin, which is a good affinity for the peptides or the proteins. So ICAT reagents come in the two types depending upon the quantity of the deuterium, uh, light and the heavy. The light, it consists the no deuterium, and the heavy, it consists the 8 deuterium. Actually, the light ICAT reagent consists of a cysteine reactive group and ICAT linker consisting of the hydrogen atoms and the biotin affinity tag. And in the heavy ICAT reagents, it is cysteine reactive group also, but it links with a heavy deuterium with a biotin tag. Now moving to the next slide, this is the principle of the ICAT. As we can see from this is the flow chart. So we can see here a new chemical reagent which is known as the isotype affinity tag is used to choose a certain kind of the proteins, and after the separations and the purification, mass spectroscopy can be used to identify the proteins. So here we can see the two types of reagents, like the first one is the light and the second one is the heavy. And after the separations and the purification, MS is used to identify the protein. The intensity ratio of a pair of ISET reagent peptide ions on the mass spectrum can be used to quantify the relative amount of parent protein in the original cells. And after that, we go for here cation exchange chromatography, and after that the LCMS to identify and quantify the ratio. Now moving to the next slide, this is the workflow of ICAT. So in from the figure 8, we can see there's a two type of samples. The first, we lyse the two cell sources in the different states and the protein is reduced. And after that, the, the two different types of different ICAT reagent is going to be labeled for the two different samples. The ICAT reagent is covalently bounded to the cysteine residue. Here we can see cysteine residue, the labeled high light ICAT, and this is the heavy ICAT of the each protein. So mix the two samples, and after completing the labeling and after that we hydrolyze them into the peptides of the different size with the trypsines. And after that, the separated proteins by the cation, we separate the protein by the cation exchange chromatography to reduce the complexity. Remove from the cation exchange chromatography, we remove all the remaining pancreatic detergents and reducing agent and the eye cat reagent also. And after that, we separated and the labeled and unlabeled peptide by the avidin uh, affinity chromatography, because we know that the benzene is a higher affinity to avidin, and the label peptides is captured by the affinity chromatography, and after that we elute and separate and the labeled peptide by liquid chromatography and analyzed by the tandem mass spectrometry. Now moving to the next slide, this is the advantages and the limitations of the ICAT. The advantages is it is useful for the protein to study the protein mixtures complexes, and it is highly automated, which allow the peptide to be sequenced directly using the tandem, and it it show the relative proteins levels between the samples estimated with the accuracy. But the limitations of this technique is that it is biased for the systems rich proteins, and it is a very expensive. This is the applications of the ICAR. It is basically used to identify and the quantitative for the cancer biomarkers. It also used to identify the component of the protein complexes and analyze their kinetics. It is used to identify the protein changes in specific subcellular fractions like the microsomes, mitochondria, and the lipid raft. It also used to analyze the post-translation modifications. So all these are the ICAT. And the next, this is the eye track technique, which means the isobaric tags labeling method. Uh, iTech was developed by Applied Biosystem Incorporations in 2004. It is a isobaric labeling method. It labels the N-terminal peptide, and it is a label-free method. Next slide. The structures and the structures of the reagent. It consists the three groups. The first one is the reporter group. In reporter group, a unique charge N-methyl piperazine reporter group consists. The second one is the neutral balance group, and the third one is the peptide reactive group. In this, it consists N hydroxy succinamide ester group that is reactive with the primary amines of peptides. Currently, two mainly used reagents, 4-plex and the 8-plex, which can be used to label all peptides from different samples and the treatment. Right. Yeah, this is the workflow of the eye track. So from figure 9, we can see it's the different types of samples. From the different sample, we extracted the protein. So basically what happened, firstly the samples are prepared under the variety of treatment settings before being lyzed to extract proteins. And after that, protein are digested with an enzyme such as a trypsin to form the proteolytic peptides. And after standard protein assay is used to assess the protein concentration of each sample. After the peptidic digestion, which is labeled with a distinct ITRAC reagent uh, like ATEX or 4PEC uh, mm, reagent before being mixed into the single sample mixtures. And after that, both for both identifications and quantifications, the mixed peptide mixtures is evaluated using the LCMS. And the tagged peptide corresponding proteins are subsequently identified by a database uh, which search utilizing the fragmentation data. And the attached tag is fragmented which gives a result in a low molecular mass reporter ion that may be used to quantify the peptides and the proteins from which they came. So this is the workflow of iTrack. The next is the advantages. So it is a very highly sensitivity and it reduces over the time and variations. Its ability to analyze the protein from the cell, tissues or the serum, and it can also detect the post translation modifications. Now the next techniques is the SILAC, which is SILAC. It means the stable isotope labeling by amino acids in cell cultures. Techniques for labeling the amino acids in cell culture medium where proteins are labeled and then detected and quantified by mass spectrometry. Uh, Actually, SILAC has emerged as a very powerful method to study cell signaling, identify and quantify relative differential changes in protein abundance, post-translation modification, such as phosphorylation, protein-protein interactions, and regulation of gene expression. It can also be used to distinguish between the proteins which is secreted by a cells in the culture and serum contaminant. This is the principles of the silence. See here, in this silex, the two types of labeling, the high isotopes and the light isotopes. The light medium compromises the natural isotopes amino acids, while the heavy medium contains the stable isotope and tagged amino acids. After the sufficient numbers of cell division, here we can see the sufficient numbers of cell divisions. All proteins produced by a cells which grown in the heavy media include a heavy amino acid. The quantifications of SILAC is based on evaluating the ratio of inserted isotope which tagged peptide to the unlabeled peptides, which is done by using the LCMS. The signal intensity from the light and the heavy samples allow for the quantitative comparisons of their relative abundances in the mixture. So this is the workflow of SILAC. Here the figure depicted the two types of uh, SILAC work presentations, like the adaptation phase and the next one is the experimental phase. In the adaptation phase, uh, the cells are grown in the light as well as the heavy SILAC medium until heavy amino acids are completely incorporated into the growing cells, which can be assessed by the mass spectrometry. And in this experimental phase, um, the two cell populations are subjected to two different treatment according to the research aim or according to the interest of mm, research researcher. Subsequently, the mixed cell populations or the protein lysate is studied and the samples are analyzed with the LC-MS analysis to identify and quantify the ratio of heavy peptided to light peptidates. So this red color is the heavy ones and the blue color shows the light ones. Upcoming to the next slide, this is the applications of the SILAC. Basically, the SILAC coupled with the LCMS and it has been widely used to characterize the protein quantitative differences between uh, different samples and to investigate the changes of protein post-translation modifications and to distinguish the specific interacting proteins in the protein-protein interactions. So there are the several applications for the SILAC. The next is the advantages of the SILAC. Uh, Basically, the SILAC is a simple and powerful method for quantitative analysis of the protein. characterized by the quantitative accuracy and it is a highly reproducibility. So now I'm going to explain the conclusion. Basically, in the last few years, the great progress has been made in the field of the proteomics, and the technologies are the very quick, sensitive, and it's going to be a good for the the proteins. So the purifications, analysis, characterizations, and the quantifications and the analysis of the bioinformatics, analysis of the huge numbers of the proteomics techniques is used in the eukaryotic as well as the prokaryotics. So this is all about the non-antibody based technology. These are all the references. That's it. Thank you.
Okay, the topic is open for discussion.
Hello.
The topic is open for discussion. So you
Yeah.
can fire your questions at Manjusha. Dr. Tarun, anything you want
Hope
to
this
comment
is
upon?
not a very boring
No,
topic.
no, sir, no question. No question.
I think she has improved since last time.
Yeah, she has. She has.
It's
Thank
still a
you.
Because
long way. It's still a long way to go.
long way. Very exhaustive topic, sir.
Yeah. Dotrina, any questions?
Questions in the sense it was a very long one. So, about ICAT, can you say once more? ICAT, like the principle.
Yeah. Can you repeat the ICAT principles again?
Yeah, Dotrina, can you please speak loud?
Dotrina wants to know the
I
principles of ICAT again.
Okay.
Yes.
So go to principles of ICAT slide.
Yes, sir. Yes, sir. Yes, sir.
Yeah, explain it.
सी दोत्रीना बेसिकली हम इसमें दो तरह के लेबलिंग का यूज करते हैं, एक
Okay.
हेवी आइसोटोप्स एंड लाइट आइसोटोप्स। दोनों मिक्सचर्स को हम साथ में मिक्स कर देते हैं और उसे हम डाइजेस्ट करते हैं विद द डिफरेंट टाइप ऑफ लाइक ट्रिप्सिन्स एंड आफ्टर दैट अब हम डाइजेशंस के बाद हम उसको कैसे अलग करेंगे कि कौन से प्रोटीन्स हेवी आइसोटोप्स कंसीडर करें और कौन से लाइट वंस। तो इसके लिए हम
Okay.
क्रोमेटोग्राफी टेक्निक्स यूज़ करते हैं
Okay.
एंड एक्सचेंज क्रोमेटोग्राफी। सो कटायन एक्सचेंज क्रोमेटोग्राफी में बेसिकली क्या होता है कि जो प्रोटीन्स, क्योंकि मैंने पहले भी एक्सप्लेन किया है कि प्रोटीन्स कंसिस्ट अ चार्ज्ड, मतलब उस पर कुछ चार्ज होंगे सब क्योंकि प्रोटीन्स एमिनो एसिड्स के बने होते हैं एंड
Yes.
एमिनो एसिड कंसिस्ट द पॉजिटिवली चार्ज, नेगेटिवली चार्ज। कुछ एमिनो एसिड्स होते हैं जो न्यूट्रल्स होते हैं जिसपे कोई चार्जेस नहीं होते हैं, राइट।
Yes.
तो जो प्रोटीन्स पॉजिटिवली चार्ज्ड होंगे वो क्या होंगे,
Mm.
नेगेटिवली की तरफ, नेगेटिव की तरफ मूव करेंगे, ठीक है। तो
Mm.
हम इसमें कटायन एक्सचेंज क्रोमेटोग्राफी का टेक्निक्स का यूज करते हैं एंड बाय यूजिंग दिस टेक्निक्स वी कैन सेपरेट द प्रोटीन्स एंड आफ्टर दैट हम इस पे जो टैग किए थे बायोटीन टैगिंग मॉलिक्यूल्स, बिकॉज द बायोटीन की बाइंडिंग एफिनिटी जो होती है वो एविडेंसेस की तरफ ज़्यादा होती है, तो वो साथ में बाइंड कर जाएंगे और फिर हम उसको अलग कर लेंगे और फिर हम उसके पिक्स को आइडेंटिफाई करेंगे थ्रू द LCMS। होप यू क्लियर
Okay.
सर, वंस यू कैन एक्सप्लेन।
Is it clear, Dotrina?
Yes, sir. To some extent.
So basically it's a three-step process.
Okay.
So the first is that you have to uh, label it, then you have to break
Yeah.
it into peptides, then
Yes.
you have to do a chromatography to separate those peptides, and
Yes.
then you have to identify those peptides using a LCMS.
Okay.
Okay,
Okay,
and
sir.
and what you are using here is an Evdin biotin reaction.
Right. Okay, sir.
Anything else? Okay, if there are no other